2 episodes back, we singled out AlphaFold 3 and IsoDDE as the technical core of this whole field, and described structure prediction as the single biggest unlock in modern drug discovery. This episode is about why that's true, and how the prediction actually works, enough to follow what the model is and isn't doing, without needing the machine learning math. Start with the problem, stated precisely. A protein is a chain of amino acids, and that chain folds up into one specific three-dimensional shape. That folded shape is what determines the protein's function and what molecules can bind to it. Now, reading the sequence, the order of the amino acids, has been easy for decades. But going from that sequence to the folded shape used to require months of painstaking physical experiment, X-ray crystallography, cryo-electron microscopy, or NMR. Predicting the shape from the sequence alone was known as the protein folding problem, and it stayed essentially open for about 50 years. That is the gap AlphaFold closed. So how did it close? AlphaFold 2, released in 2020, predicted the structure of single-chain proteins at near experimental accuracy for a huge range of proteins. That was a landmark, but limited in one important way: drugs don't act on lone proteins, they act on complexes. AlphaFold 3, open-sourced in November 2024, is the current baseline and the key generalization. It predicts complexes, a protein bound to a small molecule, proteins bound to each other, and proteins bound to nucleic acids like DNA and RNA. Architecturally, it switched to a diffusion-based module that generates the atomic coordinates directly, and that's why it can extend beyond proteins to almost any molecular assembly. And the complex is what matters, because you cannot design a molecule to fit a binding site you can't see, and the binding site only takes its true shape when the target and the ligand are considered together. Then comes IsoDDE, Isomorphic Labs Drug Design Engine, reported in February 2026. Its headline result is that it more than doubles AlphaFold 3's accuracy on protein-ligand generalization benchmarks. And the word doing all the work there is generalization. It means performance on targets and molecules unlike anything in the training set, which is the realistic condition in real drug discovery, because you're rarely working on something that's already well characterized. 2 capabilities are worth naming. First, binding affinity prediction, not just whether a molecule binds, but how strongly, which is what actually ranks one candidate against another. And second, cryptic pocket discovery, finding binding sites that aren't visible in a protein's resting shape but open up only transiently. Those are targets you literally could not see with a static experimental structure, which is how better structure prediction can expand the druggable proteome, rather than just speed up work on pockets we already knew about. Now the honest limitation, and it's a sharp one. AlphaFold 3 has documented hallucinations on intrinsically disordered regions, stretches of a protein that simply don't have a single stable folded shape. The model was trained to output a confident structure, so when it meets one of these regions, it tends to invent one anyway. This matters because those disordered regions are biologically important, many of them are involved in signaling and in disease. And it's a clean example of the generalization gap we flagged earlier in the series. The model is superb inside the well-behaved, well-represented part of biology, and unreliable outside it. Confidence scores like PLDDT do help flag the low-trust regions, but a low-confidence region is still often handed back to you as a concrete, real-looking shape. Step back, and this is the through line of the whole series in one stage. Structure prediction replaces a slow, expensive physical experiment, crystallography, taking months, with a fast, cheap computation that takes minutes. The payoff is throughput. You can predict structures for thousands of targets, and then spend precious experimental effort only confirming the few that actually matter. Plus reach, into places experiment struggles to go, like cryptic pockets and complexes that simply won't crystallize. Predict, don't measure. And keep the caveat in view: the prediction is a powerful starting point almost everywhere, and a genuinely unreliable one exactly where biology stops being well-behaved.